let's look at first your antimetabolites. And the big ones here come down to the folate pathway. Now, the folate pathway is one that is highly targeted by a lot of chemotherapeutic agents, not only in cancer, but we're also talking about antibiotics and also antiprotozoal drugs. So, three different drugs, all blocking the same enzyme in three different contexts. We have methotrexate in cancer, you have trimethoprim for bacteria, and you have pyrimethamine in protozoal treatments for things like malaria, for example. So these all block DHFR, dihydrofolate reductase, which is involved in the synthesis of folic acid, which is important for nucleic acid synthesis. So as you can imagine, if you cannot produce any folic acid, you don't have the capacity to have so many nucleotides. You need a lot of free nucleotides to build up your DNA strands. So if you have a lack of nucleotides, how can you replicate your DNA? How can the cancer grow? You're really shutting it down. Now, this also has very big effects on your normal cells, so these are very cytotoxic. That's why you have leucovorin, which is given. And what this is it can bypass this step, so you can still produce more folic acid. Or, yeah, so you could still go through the folate pathway and still synthesize new DNA bases. However, the demand of the cancer cell is so much higher than that of the normal cell. The leucovorin is really rescuing your normal cells, but just not enough for the cancers. And that's how you can selectively try to target the cancer over the normal cell. Lastly, I think we're going to talk about, so those are antimetabolites. Now, we're going to talk about antagonists of pyrimidine and purine bases of the different nucleotides. So, 5-fluorouracil is for your pyrimidines. It's inhibiting your pyrimidines. It's metabolizing three different compounds. The main which of these is FDUMP. I don't really need to know it, but it blocks thymidylate synthase, which is really important for producing thymidine to go and form your pyrimidine bases. And the other different forms of it can also get incorporated into RNA, DNA, and have a lot of more effects outside of this. So transcription and translation make it quite cytotoxic and yeah, so a lot of side effects that come with drugs like this, but also
Side effects could you mention? Okay.
yes. So well, the thing about all these cancer drugs is they're really targeting rapidly proliferating cells, all these cancer cells, especially these methotrexate, uh, five fluorouracil. They're targeting cancer cells which are rapidly proliferating. So you can imagine you're going to hit other cells which are also rapidly proliferating. Hair loss, you're killing off all your hair follicles. Um, you have a decreased white blood cell count and platelet count, so you get thrombocytopenia, bone marrow suppression. Your bone marrow is constantly proliferating. Um, and also spermatogenesis. So you get a lot of sterility as well. So these are a lot of processes. I think I just mentioned some of these antagonists, 6-mercaptopurine as well, and 6-thioglutamine. And importantly, with 6-mercaptopurine and methotrexate, the these are also used for modulating the immune system as well. Methotrexate targeting a lot of these highly proliferating immune cells can be used at immunosuppression. That's why it has a very big role and is one of the DMARDs, these modifying anti-rheumatic drugs. So it's used in rheumatoid arthritis. So finally, we have some of the plant derivative drugs. And these will come in with the cell cycle assembly checkpoint, the SAC. And you have, because when you build up, you have the spindle. You build up the spindle from microtubules. Microtubules
Yeah.
are found in usually in a dimerized form. They're free, they're floating about in dimers. And you start packing them together to form these long spindle chains to then bind to the centromere and bind to the kinetochore and pull apart your chromosomes. So two ways you can target this. You can either stop the assembly. You have,
Spindle fibers.
yep, vincristine, vinblastine, nice, and you can also stabilize the fibers as well. You can also stabilize them. So if you're able to form the fibers, but you're not able to pull apart the chromosomes, then you're stuck here. And the cell cycle assembly check will find this error and then shut down the cell cycle. So what drug do you think would help in this case?
Paclitaxel. Yeah, actually, there's a very nice story. There's a quite interesting story about Chris Christie being a governor, bad governor, blocking the roads, blocking the microtubules, Vin Christie. And paclitaxel is another drug that will be used, and the side effects, there are different types of side effects as well for these inhibitors. For vincristine, you have neurotoxicity. For paclitaxel, you have characterized neuropathy. And the final one has characterized arthralgia. That's what I know. And I can't quite recall the name. Beginning with E. Beauty.
With B.
No, it begins with E. Like Erith Beauty or something.
No,
Erybutin.
I haven't heard of this.
Vaguely remember.
Maybe one of the previous types of drugs, because I'm just going over the class of drug that it really is. Okay, so finally the last thing. So we've targeted the DNA in its in structure and integrity, but we haven't talked about how it interacts with other factors. So I mentioned with your anthracyclines, your um, mitoxantrone, and your actinomycin D, how these can sometimes inhibit your topoisomerase 2 and affect the unwinding. But other drugs, if you would like other drugs you know that block topoisomerase 2,
Right. Etoposide. Very
yeah.
typical.
So yeah, you're blocking your so those block topoisomerase 2. What about topoisomerase 1?
Topotecanate.
Yeah, so topotecan
Tecan.
and irinotecan
Yeah.
block your topoisomerase 1. So if you can't unwind your DNA, you can't really get the replicative machinery in there. Things like your beta clamp, your PCNA, can't get them in there. So yeah, in that sense, if you have these replication forks, you can't separate them out, you get stored replication forks, and therefore you cannot replicate your DNA. So that's how they really get involved. So yeah, I think we've touched on a lot of the stuff. Um, obviously, uh, very we're talking about non-selective drugs here. You're trying to get them to selectively target the cancer cells, but it's these global processes of DNA replication, DNA repair, and all of this. They're present in both the cancer and the human cells, so they will have a lot of side effects. So as I mentioned before, all the highly proliferating cells will have will start to be hit by these and have a lot of cytotoxic effects. You, yeah, therefore have a lot of severe toxicity, especially with bone marrow suppression. That's really bad because you're then liable for opportunistic infections. Also worth mentioning, a lot of these drugs can cause a lot of nausea and vomiting. Having all these chemicals in you, they act on emetic systems in the CTZ. I think that's the central trigger zone. Anyway, that's the part of the brain that's involved in emesis, and so causing a lot of nausea and vomiting. And you give it things like a danzatron, 5 serotonin, 5-HT3 antagonist, which is a very highly expressed channel in that region as an antiemetic. As a on
Okay.
the side with all of these.
That's good. I just wanted to preface earlier about the arthralgia. It's caused by something known as eribulin, which binds these microtubules. So you do have this idea of blocking microtubules. This is very common in anti-cancer drugs. Okay. All right. Is there anything we need to say? Any specific things about maybe more clinical side treatment to do with cancer? Like, I don't know actually. Like maybe apart from drugs, do you know any other effective treatments or
Well,
things that can practice?
as mentioned, your surgical approaches. So you'll often combine your chemotherapy with some form of surgery because with surgery you can eradicate a lot of the cancer, but are you really going to get every single part of it? And you have tissue around there that's normal, that's healthy. And if you're trying to remove a brain tumor, if you're removing large parts of the brain, how do you think that's going to affect the patient? So you have to have this very, you have to have this very uh,
Meticulous.
yeah, very meticulous way of trying to reduce the damage that's going to hopefully not affect the patient's life. So if you can't fully excise the tumor, so for solid tumors we're talking here, then yeah, you can use your chemotherapy as an add-on. Things like hematological malignancies, so things like leukemias, these kind of stuff, you can't target those with surgery unless you replace the whole bone marrow. But that's very serious. That's a very big procedure. So that's where your chemotherapy agents come in. Things like tumor dynamics. If you have a very solid tumor, uh, the different cells in the tumor will be proliferating at different rates. So this comes with the tumor stem cell model, is that cells will differentiate. You have these dedifferentiated cells. Some cells in the tumor will be highly proliferative, and some cells will not proliferate at all. So those cells would be highly unlikely to be hit by a lot of these drugs because they're not proliferating. They're also highly more likely to be more. So if you excise those cells, they'd be less likely to be malignant, whereas the highly proliferative ones be more likely to be malignant. Also, the blood flow in the tumor microenvironment can also alter a lot of the efficacy of these chemotherapeutic drugs. You can have a lot of drug efflux, where the cancer cell literally pumps out with things like PGP pumps all the drugs, or starts producing enzymes to metabolize them. The other thing it can do is also outcompete the drugs. If you're giving things like different like methotrexate, trying to stop these pathways of these precursors, you can start amplifying up the cell cycle and outcompete the drug itself. So there's many different and getting into the tumor microenvironment as well, physically inhibiting the drugs from getting in. So there's many things that are stopping these chemotherapeutic agents from being a mainstay. Obviously, also not selective, they're highly toxic. That's another problem that comes with that. And obviously, you have your radiotherapy, where a therapeutic approach, we're trying to target your radiation into very specific areas. Again, other issues with things like radiation and blah blah blah. So that's a important thing to take into consideration as well.
Okay. One final thing I would just mention is the idea of promoting apoptosis in certain cells. So, for example,
Oh yes, yeah.
yes, I'll just preface that by, from what I know, one of the ways you can promote apoptosis in a cell is having a low BCL2 to Bax ratio, which is two proteins involved in apoptosis. And there are many other ways you can promote apoptosis. I'll let him get into that. So, how do how can these drugs be effective in that sense? How can they promote apoptosis to kill these cancer cells? And do you have any examples as well?
Yeah, I can give you an example. So obviously you have your apoptotic pathways, your BCL to your BAX, that will then drive things like cytochrome C release, activate your caspases, and start basically uh, uh, catabolizing, breaking down your cell, breaking down membranes, releasing all these hydrolytic enzymes. Yeah. So a way that you can target these pathways again is a similar way that we were doing with these chemotherapeutic agents. What we are trying to do is induce DNA damage so that then you can start picking up the DNA damage. You have your different things like ATM kinase, ATR. They pick up things like DNA damage and they relay the signal to your pro-apoptotic proteins, things like P53, which will then interact to activate the BAX pathway. I don't know about any drugs in the BAX pathway. I don't think you can really interact with that because it's so universal. You'd literally be apoptosing all your normal cells. But what you can do is the fact that a lot of cancer cells, to survive, downregulate or they lose the function of their DNA repair machinery. So a lot of your a lot of things like homologous repair in, let's say, BRCA. BRCA is a protein that is involved, is a gene encoding a protein that is involved in homologous repair, which is repairing double-stranded DNA breaks. Very, very important for, let's say, replication forks, where you have a copy of your sister chromatid. So in DNA replication, when you have a copy, you can use that copy uh, to repair any damage or mismatch. Okay, any DNA damage. What you have, though, is in BRCA positive breast cancer, is if this is non-functional and you can't do homologous repair, but you can do single-stranded repair. You have enzymes or proteins called PARPs. PARPs are involved in I think it's more specifically MMR, mismatch repair. So if you have DNA damage and you use PARP inhibitors, your PARP inhibitors will block this enzyme. So if you have single-stranded DNA damage and you can't repair it with your PARP inhibitors and your MMR, then you have to go to double-stranded DNA techniques. So things like HR and the hedge. But if your HR is damaged in breast cancer and it's downregulated and on functional mechanism, then you cannot rectify this DNA damage. So this way, you're pushing the cancer cell to have more mutations, more non-functional proteins. The DNA machinery cannot fix anything that goes wrong. So then you're targeting again it for the cell cycle checkpoints to then not be able to pass through and the cell goes into apoptosis. The other thing as well is if you start introducing damages to the DNA, the cancer just cannot produce the proteins it requires for its own survival, then it dies as well.
Okay. Uh, I think that was,
Sufficient?
yeah,
Comprehensive enough?
a good deep dive into all the secrets about cancer, anti-cancer drugs, apoptosis, promoting all the inhibition of all the paths of the cell cycle, the standard factors, um, the thing you mentioned at the beginning,
Hallmarks of cancer?
hallmarks of cancer, and everything under the sun about cancer. So now you can approach any exams you have, any understanding you have of cancer with new mind, hopefully. Hopefully you take away
New
a
mind,
lot
new
from these
soul.
episodes, new soul.
Yeah.
And stay tuned for our other videos in the Nerves Breakdown podcast. We break down biology, even A-level biology, even AS biology, even medicine, chemistry, everything alike. So, thank you for tuning in to the Nerves Breakdown podcast.
And please let us know what other videos we can do for you guys.
Absolutely. We'll see you in a minute.